In this lecture, we will be discussing how biomimetic stochastic topography and electric fields synergistically enhance directional migration of corneal epithelial cells in an MMP3-dependent manner. The anterior corneal surface is covered by a stratified epithelial layer that is associated with a rich 3D, topographically patterned specialization of the extracellular matrix. Primary functions of the corneal epithelium include protecting the eye from external physical, chemical, and biological irritants, and providing a barrier to microbial invasion by maintaining a protective junctional barrier. Wounding of the corneal epithelium results in a loss of barrier functions. Directional cell migration of epithelial cells is critical in wound healing. Previous work includes measuring the bioelectric fields in mice and human skin. This was found to be around the order of 177 millivolts per millimeter. In this particular experiment, the max electric field strength induced was 150 millivolts per millimeter. Gao and his colleagues also measured the corneal wound current in rats. This was found to be 4.3 microamps per centimeter squared ex vivo, while it was 24.8 microamps per centimeter squared in vivo. This experiment presents three main factors in regulating cell migration. These include surface topography, substratum stiffness, and induced electric fields. These two images show the experimental topographic setup. Image A shows an atomic force microscopy height mode image. And the graph on the bottom shows the cross-sectional height of the topographic features along the blue line. Figure B is a scanning electron microscope image of the basement membrane of a rhesus macaque, inferior vena cava. This was used as a baseline for measuring physiological relevance of artificial extracellular matrix. This figure presents the experimental setup for the testing of cell migration. There is an electrotaxis chamber with biomimetic stochastic topography placed within a petri dish filled with culture medium. A voltage can then be induced across the medium. A PDNS stopper was first placed on the topography and cells were seated around it. The stopper was then removed in order to simulate an artificial wound. This figure shows a directional measurement of cell migration in different electric fields. These images reveal that inducing an electric voltage stimulated cell migration towards the cathode. The cells migrated further under a stronger electric field. These microscopic images show that cells aligned more in the direction of the magnetic field when placed on stochastic topography. While the cells on the flat topography still migrated towards the cathode, they did not migrate in as straight of a line. Graph A compares directedness of cell migration in comparison to induced electric field. Directedness of cell migration increased as the electric field strength increased. But when comparing the flat versus stochastic topography, there was no significant difference. For graph B, when comparing track speed with electric field strength, at 115 millivolts per millimeter, cell migration speed increased drastically in the stochastic topography compared to the flat topography. This image compares the track migration speed of a single cell on flat versus stochastic topography in the presence of an electric field. As you can see, the cell on stochastic topography has migrated further than the one on the flat topography.
Alright, so today I'll be talking about the upregulated expression and activity of MMP3. It was observed in the study that in the presence of an electric field of sufficient strength, there was a marked increase in MMP3 mRNA production in contrast to the two controls, which were MMP2 and MMP9. In the following figure, you can see that MMP3 mRNA expression in the presence of an electric field was disproportionately higher in cells grown on a stochastic surface. In cells which experience high electric fields and subsequent increases in an MMP3 upregulation, the absence of any increased levels in the supernatant suggests minimal excretion of MMP3. And treatment of MMP3 inhibitor was also shown to not significantly impair expression or excretion in any affected cells. The following figure here presents a fluorescent labeling assay which further confirms the increased presence of intracellular MMP3. This following figure depicts forward single-cell migration on planar and stochastic surfaces. These results imply that stochastic surfaces enable cell migration as uniform sheets, as opposed to sporadic single-cell migration. These two figures here depict single-cell migration trends in planar and stochastic surfaces. As you can see, there's significantly more spreading of all the cells grown on planar surfaces that results in dissociated cell migration, and effectively this will translate overall to less efficient wound healing. In the open field view microscope images provided here, you can see quite the opposite was the case with stochastic surfaces. The cells move more uniformly as a sheet into the artificial wound, and this likely will represent an overall more effective wound healing process.